So let me start off then by doing an introduction to our first speaker. It's Dr. Thomas Garcia. He earned his PhD in molecular and integrative physiology at the University of Illinois before doing a postdoc at the University of Texas MD Anderson Cancer Center. Currently, Dr. Garcia is an assistant professor at the Baylor College of Medicine in both the Department of Pathology and Immunology, as well as the Scott Department of Urology. His research focuses on spermatogenesis and male reproductive system. In recent publication, Dr. Garcia and his colleagues have reported on a number of genes involved in male reproductive tract, some of which might be potential targets for male contraceptive therapies. Expanding on this research has led to reviving an important tool for the reproductive biology community. And today we'll get to hear more about this work on the Mammalian Reproductive Genetics Database, MRGD version 2. So take it away. Thank you.
Thank you, Anna. Thank you so much. It's a pleasure to be here today to talk to you about the work that I'm undergoing in my research lab. And let me go ahead and share the slide. And I think this is it. All right. Do you guys see my presentation?
It looks perfect. Thank you.
How about
yes.
now?
Perfect.
Full screen?
Perfect.
Okay, great.
Yes.
Yeah. So to give you a little bit of a background, I'm part of the Center for Drug Discovery at Baylor College of Medicine, and this Center for Drug Discovery is very interested in identifying novel, non-hormonal male contraceptive drugs. And so, because currently there is not a orally bioavailable male contraceptive, and it's a big problem. There's a lot of oral contraceptives for females, but there's not a single one for men. There's been some progress in terms of the hormonal methods for men, but there's side effects, just like there's side effects in women. And so what we need to do is identify proteins that are specific to the male reproductive tract that are required for male fertility. And so what this shows is how our drug discovery pipeline works. And so we start with the informatics, transcriptomics, and proteomics to identify genes that are restricted in expression to the male reproductive tract, to the testis or the epididymis, for example, right? So this would be a representative of the kind of target that we'd be interested in. We'd also be interested in identifying druggable genes, and this is where Pharos comes in, and illuminating the druggable genome. And a while back, when I first started my work at Baylor College of Medicine, I was made aware of the illuminating the druggable genome, and I took full advantage of the database that was available at the time. And I still use it to this day. There was an individual at the NIH, part of the consortium, that I contacted. He was very generous and helped me to provide lookup tables that I use to this day, Dactrum and Wnt. And so that involves identifying the druggable genes, the druggable proteins, the kinases, GPCRs, ion channels, as Anna had mentioned. And so after we identify genes that appear to be reproductive tract specific through the high throughput transcriptomics and proteomics approaches across reproductive and non-reproductive tissues, we have to verify the expression is restricted through RT-PCR. Now, one of the steps that we do in our group, that I carry out in my lab, is generate novel knockouts. So not every gene has been knocked out yet. And when we identify a gene that's restricted in expression, and especially those that are potentially druggable, kinases and other enzymes, then we're very interested in seeing what happens when we delete that gene. And so I've generated quite a number of knockouts since I started at Baylor College of Medicine to determine whether or not it results in an infertility phenotype. Now, if the animal is subfertile or has no phenotype whatsoever, then we're not interested. That's a no-go situation. The burden of proof for a male contraceptive is high. It has to be highly efficacious. It has to work, and there can't be any side effects. And hence the reason why the target should be restricted in expression. And even paralogs should be very dissimilar So there shouldn't be off-target effects. If we identify a drug compound that binds to our target of interest, we really don't want that drug to bind to something else that has sequence similarity. So we also do that too. I carry out those searches to make sure that the percent similarity, especially in the druggable regions, are low with the closest paralog. So we identify the target of interest, and now it's getting to the point where a lot of the colleagues that I work with in the Center of Drug Discovery, they take over from there. I do protein purification in my lab, expression and purification, but then the drug library screening that's part of the Center for Drug Discovery, we use what I believe to be a pretty innovative and novel approach. We use DNA-encoded chemical libraries. So instead of having one molecule per well in screening molecules that, at that rate, we can really only screen in the tens of thousands or at most a million different compounds. We have libraries that are in the billions of distinct molecules. And each of those molecules is ligated to a unique barcode that identifies what the molecule is. So what we can do is we can do pull down with, the protein is affinity tagged. We do a pull down, and then we see which molecules are stuck to the protein through DNA sequencing. And so once we identify the highest binders, then we have to build them off of the DNA scaffold. So it's hit resynthesis, and then study the pharmacokinetics of the compound and the effect that it has on the protein. And then, of course, then we're at the point where we want to see whether or not the drug has the effect that we would expect it to have if it inhibits an enzyme that's required for fertility. And so we have a lot of different candidates that we've moved forward with that are pretty far along. This process works, but I'm going to focus on the area over here. So in 2017, I was made aware of an RFA that was available through the Contraceptive Research Branch at the NICHD. And the purpose, well, to give you a little bit of background on that, the program officer at the NIH, a decade before, had done a number of microarray studies to identify male reproductive tract-specific genes, testis-specific genes, and epididymis-specific genes. And so that was his baby. And so now, 10 years later, he's a program officer at the NIH, and he's aware of the fact that the field has moved significantly in the last decade, and it's no longer microarrays that dominate high-throughput transcriptomics, but instead it's RNA-seq. So he put out this RFA to solicit proposals to try to, as he put it, to me, to drain the swamp, to try to identify more targets than had been identified before. Anything that had been left undiscovered, he wanted it to be discovered for the drug development community to be able to move forward with if it's a viable candidate as a male contraceptive target. And so I submitted my proposal to perform transcriptomic analysis of human epididymis segments and testis to identify novel targets to interfere with sperm maturation, perform proteomic analysis of human sperm. And then, in fact, I threw in an extra aim, which wasn't part of the RFA, but I said I was going to validate these targets through knockouts, because that's what I really do. I'm a male reproductive biologist by training. I really enjoy knocking out genes and studying the phenotype in animals. So I put that in, and my proposal was funded. And so the main paper that resulted from this work, it's already been published, and so you can look it up and you can find it. It's got a tremendous number of supplemental figures and data sets in there. All the data that I'm going to show you is in this public, well, not all the data, all the male data that I'm going to show you is in this publication. I'm also going to show you basically identical work that I've carried out since then for female, and with about 4 times the amount of data, and so that I can put it on this website. And that's what I'm going to showcase towards the end. But basically, this is the process. A large number of non-reproductive tissues, lung, liver, adrenal glands, stomach, duodenum, came from one study. And I don't think I have the reference here for that study. Oh, here it is. That's the one paper where I got all the RNA-seq data sets for the non-reproductive tissues and for testes. Now, in order to identify all the undiscovered targets, I wanted to dive deeper, and I wanted to look at RNA-seq data sets that were publicly available, but had not been used to do a side-by-side comparison with non-reproductive tissues to identify genes that are specifically expressed in those cells or other tissues. So I harvested data sets that were available at the time for spermatogonia, spermatocytes, and spermatids from 2 different studies, Sertoli cells from 2 different studies. One of them was really good, GFP positive under the SOX9 promoter. They were FACS sorted, purified RNA-seq on bulk RNA-seq on those. And then for epididymis, there had been one paper where they did an N of 2 for segment, the Caput corpus incauda. But I contributed 3 new data sets to the community. Those are deposited in GEO for Caput corpus incauda. I also added another 3 data sets for testis. Okay, so those are so this is human, and this is mouse. So actually, these were in the SOX9 GFP. The SOX9 GFP were here in the mouse, and so same situation. The non-reproductive tissues were harvested from one study, and then the reproductive tissues came from a variety of different sources. But again, those sources did not analyze those RNA-seq data sets in the context of trying to identify genes that were specifically expressed. So I started with all the raw data and ran it through the same pipeline in parallel in order to be able to normalize the data, in order to be able to look at the side-by-side expression across all these different tissues and these different cell types. And so in here, there had not been any RNA-seq done on mouse epididymis. There had been for human with the N of 2, but I increased that number, and then again, there had not been any epididymis RNA-seq. There had been microarray, like I said, but I contributed that, and that's deposited in GEO as well. So then the result is there were a lot of genes that I identified through all of these RNA-seq data sets. There were 263 RNA-seq data sets in total for human and mouse. And of those, there were over 1,000 genes, after all the filtering that we've done, over 1,000 genes that were reproductive tract specific in the mouse. There were just under 1,000, 720 that were reproductive tract specific in human. And then the big question is, how do they compare if they're orthologs? And so how many of those overlap? But I wanted to be able to parse this data in terms of, yes, indeed, whether or not there's an ortholog, how many orthologs there are, whether it was novel and previously identified through other, like I mentioned, the microarray-based high throughput studies. There was one RNA-seq study, but only testis, not epididymis. And so in terms of novelty, how many of these targets had been previously identified? And so for mice, for previously published mouse or rat data, I figured out what the orthologs were in human, and I considered anything that was an ortholog previously identified as well. And so what you're seeing here is truly for which, when I say there was 346 genes, human genes, with a mouse ortholog, for which they had not been previously identified, that includes any gene that was previously identified in the other species, for which we can consider the ortholog in the human to be previously identified too. And so 346 human genes for which there is a mouse ortholog that had not been previously identified. And next question is, has there been a mouse model made? Even though they had not been identified in high throughput transcriptomic studies, these genes may have still have been studied and mice made, and those mice may have had infertility phenotypes. And indeed, of the 346 genes, 113 have knockouts, 86 of which have no phenotype or are subfertile, 27 of which have infertility phenotypes. So these are actually potentially viable drug candidates. And then over here, 233 genes have no mouse model. These are of interest to me because I like to knock out genes and then see whether or not they're dispensable or required for fertility. And on this side of the map, again, for the mouse gene, same question. Do they have human orthologs? Were they previously identified or novel? Were knockouts previously made? If they were, how many of those were subfertile, dispensable, or resulted in infertility, male infertility? How many of them had, how many of these genes have no mouse model? And then when we compare these in terms of orthologs, how many of these genes are identified as reproductive tract specific through this work that is reproductive tract specific in both species? So about 109 genes are novel without a mouse model. And so here's a glimpse of what those genes look like. And then this is where Pharos and Illuminating the Druggable Genome comes in, because we'd like to parse it further to be able to identify genes that could become potential drug candidates. And that doesn't mean that a protein that's not considered an enzyme, kinase, GPCR, or otherwise couldn't be a drug target. If we were to prioritize these genes, we would certainly want to prioritize them based on these categories right here. And so parse out over here, you can see like a metalloproteinase and solute transporter, olfactory receptors. If for some interesting reason, there's a lot of olfactory receptors that are orphan genes, olfactory receptors that are expressed in spermatogonia and in sperm. Very interesting. Proteases like the TMPRSS and PRSS genes, and then there's a number of these WFDC and QNITS domain-containing proteins that are expressed in the epididymis. And so in this Venn diagram, what I'm really trying to highlight is there were genes, novel genes identified from the testis data sets, but you can see there was a lot of wealth added in terms of novel genes identified by introducing the spermatogonia data sets and the spermatid data sets. And I believe if it wasn't for introduction of those, we wouldn't have been able to identify these genes as reproductive tract specific in the testis. And we wouldn't have identified these if we didn't introduce these as well. The new data sets, the human epididymis data sets. So this is what the data looks like. This is the way that we parse the data. You can look at it in terms of just the mouse genes that were identified. Similar situation. There were stem cells, spermatogonial stem cells using ID4 GFP positive cells, and like I said, SOX9 GFP positive Sertoli cells, fact sorted to give us these genes that we would have otherwise have overlooked if we didn't include those data sets. And we have some overlap with the testis data sets. We still identified a fair number of genes as reproductive tract specific, and also underlined here are genes, like I said, the ones that they're reproductive tract specific in both species. So you should actually see the 109 genes here as well as over here as underlined in both of these Venn diagrams. And so we can go further. Now, these don't have mouse orthologs, so they could be of interest. There are some druggable ones. These genes, this gene, potentially druggable, but the situation is such that we can't knock it out in a rodent model. So if we were to try to study this or move forward with it as a drug candidate, we wouldn't have that validation data showing that the absence of that gene results in an infertility phenotype. And so 142 previously identified human genes without mouse models. So we still, even though these had been identified previously over a decade ago, we still don't have a mouse model for them. And this is what I like to present the data as. So I call it a digital PCR, but it basically is a heat map in PCR graphical format that depicts the expression level, transcripts per million expression level. So if the transcripts per million is 30 or more, it could be 3,000, 1,500, or 30, it'll be black, because that just means it's expressed strongly. And anything between 0 and 30 is some shade of gray, so 0 would be white. And what I want to highlight here is these are the non-reproductive tissues, and for all of these genes identified, you can see that the transcript per million values are essentially down towards 0. And same thing for the mouse. And we spent a considerable amount of effort trying to verify these through RT-PCR, and we can verify them. for a number of the genes that we tested. You can see it's restricted in expression. And now here's where I want to fast forward to what we've done more recently. I showed you the male human and male mouse, but then the study was repeated for female, including, and if you notice, the number of studies for the male, 7 separate studies, 5 separate studies, with 171 and 90 data sets combined. For female, a couple of years went by, not 2 years, but between when I did the male and when I did the female, more RNA-seq data sets became publicly available, and the female also has a lot more moving parts to it. So it's not just testis and epididymis, but now we have a lot of different. We have the uterus and various layers of the uterus, endometrium and myometrium, placenta, mammary gland. I even included vagina for human through dbGaP data sets, included zygote, different stages of development, different ovary cells, granulosa cells, cumulus cells, and different oocytes, different stages in ovary. And so a lot more data sets, 450 data sets from 19 studies for the female human, and for mouse, female mouse, 318 data sets. And so what I'm going to do is this is, again, the graphical output example of what the data looks like, with some representative genes. This is a granulosa cell, or the ovary-specific gene, oocyte-specific gene, and so the embryo-specific gene, the progesterone receptor, which you find very strongly expressed in the uterus and endometrium. So all of this makes sense, and it's the data. It pretty much makes sense between both species, from human and mouse. So what I'm going to do is I'm going to show you. This was the previous MRGD website that was initially made in 1999 and only hosted microarray data and hadn't been updated since about 2000. So, 2010. Around 2010, 2011, that's when this, 10 years ago, this website ceased to have new data sets added to it, and the genes, the nomenclature for the genes hadn't been updated. And so a lot of the microarray data sets were hosted here, but not the RNA-seq data sets. So this is where the Male Contraceptive Initiative stepped in, provided me funds in order to develop this website, the Mammalian Reproductive Genetics Database. And so this is a screenshot of what it looks like. And I think what I'll do is I'm going to just show you a live demonstration of what the website looks like. And then, in fact, if anybody has a gene of interest that they're interested in me demonstrating, in fact, that would probably be better if somebody could just shout out the name of the gene symbol. Hopefully it's the current gene symbol, and then I'll show you what this looks like. Does anybody have a gene that they would like me to look up?
We'll need you to add it into the chat for the.
Oh, great.
KLK3 and GPER1.
KLK3 I'm familiar with. It's a kallikrein in the prostate, so I don't have that tissue. So KLK3, let's just see what it looks like. It's a human. There's no mouse ortholog. View charts, male. And so let's see what it looks like. Oh, there is prostate. Maybe I'll change this to minimum value because it works great for, oh, there's a lot of orthologs. Here we go. Okay, view charts. So the way that this works is there's an ortholog lookup table. So if I enter a human gene symbol, that's this human gene for KLK3, then the mouse ortholog will pop up. And so apparently there's a number of mouse orthologs, and correct me if this is incorrect, but this is what the lookup table is telling us, is that this one human gene corresponds to all these different mouse orthologs. So the website is designed to show you all the orthologs that the gene corresponds to. So this is RNA-seq data. One of the revisions that I'm going to make, and again, this is still under development. One of the revisions is I'm going to revise this so that it states precisely that this is RNA-seq data. This is in fact the data that I've been showing. You can click on this and it will show you the SRA values for the raw RNA-seq data sets that the data was derived from, shows the citation, number of samples. I'm going to do that over here. It's a different study. This is the Human Protein Atlas data. Do that for this one. You can see all of the SRA values for the data. Okay, and now I also at the request of the program officer at the NIH, who had all this data published over a decade ago, he asked that I introduce this former microarray data onto the website as well. And so you can see what the data expression looks like. This needs to be corrected. I see if I hover over here, you can see that it'll tell you what the TPM value is. It's very highly expressed in the prostate in the human, 5,904 transcripts per million. So that's accurate. That's accurate. Unfortunately, the web developers, they call this TPM, but it's not. This is microarray data, so it's not transcripts per million. So that needs to be fixed. And then there's bar charts. And all of this is customizable insofar that you can pick. So for the, we can change the order. You can drag these up or down. We can exclude them or include them. We can have them in the chart, not the digital PCR or heat map, or vice versa. So I won't go through all the various permutations, but you can see that the way that we designed this website, the user should be able to generate a graphical image of use in one, whether a bar graph or otherwise, and then be able to actually download this data too. So there's an option here for save charts as image. So we can click on that, and it's going to zip everything into a folder. So for PNG files and other file formats. So hopefully this is going to be helpful to the research community. And I think I'm over my time. I have to hand the mic over to the next group. So if there's time, or if anybody wants to shoot me over any questions or anything like that, I can answer any other questions that you guys have. But I don't want to take up any more of the time.
Okay.
Thank you.
Maybe we can come back at the end. Thank you very much, Dr. Garcia. I appreciate it.
Thank you.
So so now we're gonna head and turn over to hear some work of the IDG informatics tools, namely Pharos, which is the user interface for the IDG's Knowledge Management Center. This presentation will be made by two key developers of this project, Timothy Sheils, who is a senior application developer at NCATS, who has been involved with Pharos since 2018, along with a long list of other NCATS projects, and Dr. Keith Kelleher, who's a senior full stack developer at NCATS and also has a PhD in biology and neuroscience from the University of Houston. Together, they will be presenting on Pharos, an open data resource for examining target, disease, and ligand interactions. And once again, please post questions and in the Q&A or chat box. And thanks. Take it over, Tim.
All right. Thank you, everyone. Thank you, Anna. Go ahead and share my screen. All right. So that should have everything out of the way. I actually, for once, have more slides than doing a live demo. Usually, I am in favor of the live demo, but I figure it'd be good to kind of build these around some of the use cases that we've recently started adding to Pharos. So I'm going to talk about, like Anna said, Pharos, which is an open data resource. We've actually kind of expanded more to include more tools and analysis, but the general idea is it's to examine target disease and ligand interactions and how all of these are interrelated and can work towards or against each other. I'll throw the website up here real quick. It's pharos.nih.gov for anybody who wants to follow along or click around while I'm going through stuff. Pharos is part of the Illuminating the Druggable Genome, which is an NIH Common Fund program. And the goal was to improve understanding of properties and functions of proteins that are currently unannotated and understudied. And NCATS, which Keith and I are a part of, our focus is on Pharos, which is the front-end user face to browse the TCRD. TCRD is a database that aggregates, depending on our counts, 78 79, 80 different data resources, from expression data, GWAS studies, ligand and drug interactions, phenotypes, target-target interactions. And we take all this information, stitch them together into common entities, and try to rank and for the targets. We come up with the target development level based on the amounts of knowledge that are available for each target. So we have T-dark, where little information is known. T-bio, there's some biological functions known. T-chem, there's some chemical activity, and T-clin has approved drugs. And there's different cutoffs and parameters that are used to rank this, and you can examine those for each target details page. So you can kind of see where it is in the pipeline, what parameters it's already met, what it's still working towards, and it gives you kind of an overall view of how much knowledge is available about a specific target. So within Pharos, it's the interface for this Target Central database. Currently, it contains a little over 20,000 targets. Actually, we've slimmed it down now, so fewer diseases as we've improved our ontologies, and a lot of ligands, so 267,000 ligands. With all of this data, we have over 40 filters, probably tons more, of just different ways you can batch and group all of this data to examine it. And a newer function that's been added that actually Dr. Garcia would have been very useful is you can upload and save custom lists. So when you have your list of targets you're interested in, you have that overlap of targets and data sets, you can upload it and save it in Pharos. And with the increase of analysis tools that we've recently added, you can really examine your data set and try to find more commonalities and more overlap. So current efforts within Pharos involve persistently enhancing the usability of the app. We're always giving demos and trying new visualizations for data, new ways to show it, adding new data sets, streamlining data sets. So trying to keep this as a living resource to be able to keep expanding the knowledge available. So the IDG program also has groups that are generating data. So we're trying to incorporate that data and tools that are generated by those. And then within NCATS Informatics, there's other tools and initiatives that have some overlap that we try to include as well. So Pharos itself, you know, I've mentioned this already, but it's designed to search, browse, and filter the TCRD. We've got a lot of different visualizations based on the data. We've got donut or pie charts to visualize filters. We've got upset plots to actually be able to view overlap between some of your data sets. And this is really designed to kind of help users identify patterns and summarize their results, to be able to see kind of from a broad picture of if there's a lot of targets that have the same properties, that's something that could be of interest. You can also access the raw data either from downloading the TCRD database itself, or you can download data from each page, just the data sets that you're interested in. We've recently switched to GraphQL for our API, which to provide programmatic access. So you can really fine tune the queries that you're running as a developer and just get data that you're interested in. And that way you can incorporate this information with other applications internally or externally. It makes it really easy to just, yeah, if you just want the TDL level of a target, you can fetch that pretty easily using GraphQL. So I want to go over some more specific use cases instead of focusing on some of the aspects of details pages, more of what you can do with Pharos. And a lot of this is based on newer developments over the past probably year or so in newer versions of Pharos. So starting off with some of the basics of searching for target disease or ligand, we've got our search box, which is always a work in progress. When you're searching, you know, do you search based on name, other instances of the word appearing? Do you search just targets, just ligands, just diseases? And so we've always been wrestling with how to do those searches, whether you nest them or do everything at once. So we've recently improved our search functionality, so you can search targets, diseases, drugs, and ligands. You can also search target family, genes, gene ontology functions, processes, viruses that may interact with them, different pathways. And we've gone from this 3-level search of you could search all of the targets, all the diseases, all the ligands, to being able to find the hit that it's a ligand or it's a target, or find different list analysis. So you can find diseases associated with this target or ligands associated with this target, or if it was just a pathway or a family, it can show you that information as well. And what that will result in is a visualization of the matches for that specific query. So you can see there's 75 targets with that query, or one ligand if that's what you were after, or the diseases associated with it. So even from this basic search for one term, you can kind of see all the different levels that it's involved in, all the different places it could have shown up. And instead of just showing a list of targets that you may not have been interested in, it's useful to be able to show kind of where everything's hitting. And then especially for some of these filters, to be able to find filters and different properties, different ways you can bundle and view this data. Another increased functionality is being able to find similar targets. And with all of these different filters, it's useful to know what other targets share that property with the target you're looking at. So for example, if we have GPR68 and we're interested in finding targets that interact with this target, it's much simpler now to be able to just press a button and it'll take all of the targets that interact with your starting target, and you can view them, see what the details are, and you can take this list and filter this list and run different analysis on it based on kind of a subset of targets that are related to your initial target of interest. So you can find commonalities or differences based on the full list. And throughout Pharos, we've got it divided into a table view, which will show you kind of the list, the basic data list of all the targets, and then list analysis that lets you perform a bit more functionality associated with it. And in the table view, once we have new information, it could be interaction details or target overlap, or even ligand prediction, that shows up as new filterable columns as well. So there's a lot of ability to go from a target details page to a bigger list of targets that share that functionality. And then the idea is you can use that list to analyze and filter down and hopefully get more information about the target you're working with, what its functionalities are, and hopefully maybe some darker targets of interest that don't have as much knowledge available for them. So I mentioned earlier finding similar targets. So if you want to find targets that are similar to DRD2 that share the same Go process, I filtered these by T-dark, so ones that don't have a lot of information. You can kind of see what the similarity is between some of these, between the list of Go processes for this target. So out of 421 targets that have the same process, or I guess it was more than that, it's slimmed down to 421 darker targets that have some sort of overlap. So you can find ones that have more commonalities compared to other. And again, based on starting with a well-known target of DRD2, you're able to find some of these darker targets that may have some potential druggability associated with them. We've also added filter value enrichment. So again, starting off with the jumping point of interacting targets, or you could do this with disease association or ligand activities. You would go from here onto a details page. This is still DRD2, and you can click this to explore interacting targets. And it'll bring you back to the regular list page, but there's functionality added to be able to calculate enrichment of filtered lists, or to calculate the scores, see which of these, there's 8,000 of those other targets associated with ovarian cancer that's associated with DRD2. But by figuring out how much that's overrepresented, we're able to cut out a lot of the noise and find targets that are actually more better suited for the disease that's better suited for this target that has more relevance to it. And then you can find these 206 targets also associated with psoriasis. That's also useful if you take all of the diseases associated with DRD2. Again, that tends to, there's a lot of the cancers that show up because of a lot of targets are associated with them. So running this filter analysis based just on diseases associated with DRD2, you filter out a lot of that noise and start to find some of these depressive disorders, cocaine dependency, that's more associated with DRD2. And you can kind of see the amount of what the expected frequency and the amount of likelihood that it's not extra noise within this list. We also have added more structure search and ligand search capability. A very early version of Pharos had structure search, but we've been able to bring that back in. And if you were starting off with a structure, you may have a CAS number or a SMILES or just a name. We've hooked this up to a tool we've developed at NCATS called NCATS Find that can then resolve this name or a SMILE or a uni and pull in the structure for you. And this is the Marvin sketcher from Chemaxon. So you a lot of people are familiar with this, and you're able to upload images, download images, save them, or you can draw a structure, whatever you're interested in putting in. And from here, you can kind of split off into two directions. You can just find similar structures based on the structure itself, or you can predict targets that may also share an activity, and that's based on the NCATS predictor tool to be able to look at the data sets for that. So starting off with just similarity search, you get a list of all of the ligands that are associated with the structure and what the similarity is. And we've got a lower Tanimoto score of 60 just to bring in as many as possible. From there, again, it's a matter of slicing and dicing it using the different filters and list analysis available. So you can filter out based on whether it's an approved drug or an experimental ligand, and the number of targets it has activity for, how many of them are unique. And again, you can really find a lot of similar ligands to the one you're looking at, and from there find targets that the original ligand you're querying may also be active against. So I mentioned the filters. So if it's there's a lot of the same target associated with all of these similar ligands, it may be a useful ligand to try to target with your ligand. Same with the Panther class. If there's a lot of receptors, it may be useful for that. Or you can slim down. There's one target that has 14 ligands that are active with it, and a lot of them just have one. So you can really refine your results and use that as kind of a jumping off or even a continuation point. So yeah, that's basically what that entails. For prediction, we actually have targets that are, there's either predicted activity. If it's known, we can find nearest activity based on if a compound is matching. And this is running through the NCATS predictor API, which will then also return what the activity is, where it might be associated, and what the model itself was. So you're able to kind of run some of this analysis on your own. And again, it's going from a ligand to a list of targets that may be of interest. Moving over towards darker targets, which kind of one of the goals of the IDG program is to illuminate the druggable genome. So to find more information or to emphasize some of the darker targets that are available. So if you have a dark target that you're studying or you're interested in, we kind of have three different directions you can go to hypothesize some of the functionality associated with it. I've mentioned earlier, a lot of the sections of the Pharos application have jumping off points to be able to find similar targets based on UniProt keyword, phenotype, disease associations, active ligands. So that's a good jumping off point there to find other ones that have the same filter. Same with protein-to-protein interactions. You can find all the targets that are known to interact with this dark target and hopefully see some patterns and some commonality there. A third method that we recently added was running a BLAST-P search based on the protein sequence. And we've got a detailed sequence viewer on a target details page. So if the sequence is available there, you're able to populate this search query for it and explore matching targets, and you'll get a list of targets that match, and then how much of an overlap there is, the percentage of the identity, and you can click on each of these lines to see kind of what the data return from BLAST-P is. So again, starting from a dark target, you're now getting a list of targets of interest, and from here, using this list analysis, you can generate heat maps or lists based on these the diseases that all of these targets are associated with, what ligands are active against them, and what other targets they interact with. So again, that's kind of a way to help you hypothesize of what functionality this dark target may have. On the other end, you may be interested in finding a dark target to study. So we've heard from some people there's IDG lists of interests, and some of these are darker targets that they're interested in studying, and they may luck out, they may already be studying that target. But if there's if you're studying a well-known target or a better known target, and you're interested in finding some of these dark targets to study, there's a lot of different ways you can do that. Again, it's based on a starting off point, a jumping point. So if you're starting off with Pharos, you can, and if you want to find understudied ion channels, you're interested in the nervous system and you're interested in IDG funding for them. So from the home page, there's filters on the side of the screen. You can select all the ion channels that are listed. So already you're going from 20,000 targets down to 300 some. You can search for nervous system phenotype and then refine the list based on the IDG target list. So you've pretty quickly, with just 3 clicks, gone from 20,000 targets down to 13, and you can then find darker targets that may be of interest and have IDG funding. We actually just released an updated IDG list, so I'll talk about a little bit more about that in a few minutes as well. Some other examples, if you're studying a specific target, we have CMKK2 as an example, and you're able to jump to different sections based on information known about each target. And again, we're using this protein-to-protein interaction as a jumping off point. So you're taking your known target, finding all the targets that interact with it, which then brings you to a new page of 65 targets that interact with your starting target. From there, you can filter ones that are T-clin, and I mentioned earlier, these are ones with approved drugs. And from here, you can filter out just the 10 targets with approved drugs, which may also, some of the ligands, some of these drugs may also be active on the target you're starting with. And I mentioned again earlier, this dynamic content based on what you're interested in. So here it's got the interaction details from the protein-to-protein interaction, where it comes from, and the strength of that interaction. And then finally, we've been talking a lot about ligands, we've been talking a lot about the targets, but if you wanted to start with a disease of interest, if you were going disease specific, you could search for asthma, find this is top level asthma, but we also do have the hierarchy available of some of the parent terms and child terms if you wanted to refine or broaden your search. We list targets again associated with that, some of the descriptions and identifiers associated with that disease, and you can get 470 targets that are associated with asthma. From there, you can filter by the data source. So if you just want ones from the expression atlas, then you can sort it by the log2 fold change to figure out how relevant they are. So, you know, that'll show you kind of what the expression level is for that gene between somebody who has the disease and somebody who doesn't. So you can filter based on that fold change and then try to find targets that could be significant with your disease. So wrapping things up, you know, there's a lot going on with Pharos. It's changing frequently as we add data sources, but we've really been focusing on moving from just displaying data that's in TCRD to helping users analyze what's in it and what can be in their data sets, to be able to upload their data sets and find information associated with that. And we've always had this idea, this focus on serendipitous browsing, of being able to just start with the jumping off point and kind of click around and find different ideas, different targets that you may be less familiar with. And again, with the emphasis from the IDG program and the Common Fund, hopefully that can help drive research to darker targets. And in addition, there are resources from the IDG available. There's targets of interest within the IDG, and there's resources generated by some of the different programs of the IDG. So there can be mouse lines, genetic constructs, mouse imaging, and when that's available, you're able to filter lists of targets based on those as well. So there is new data being generated by the IDG and resources available for your use. So we're always also looking for feedback. Is there data that you think should be added? Relationships, anything not working? Are you interested in a more in-depth walkthrough or training? We're available to do all of that. We've got the website here, an email address. You can contact myself or Keith. We're on Twitter, and on the site itself, there's a feedback button where you're able to, you know, let us know if you see any issues or have any questions. We've got a long list of acknowledgements. It's always changing, but it's a large group. It's a large collaboration. A lot of people have been working on this this program, and even within NCATS, we have a research core grant as well. That's funding a lot of this and keeping Keith and myself busy. So I'm going to end this with a picture from a hackathon we did a long time ago, I guess 2.5, 3 years ago. So hopefully one day we get to meet in person again, and I'll probably have to visit a barber before I go down there. So any questions, feel free to throw them in the chat, and I'll turn it back over to Anna. Thank you.